Hello, everybody. Welcome to Biology Round 2. Oh, you guys hopefully did really good on that first chapter, Chapter 6, with all those sections. Don't forget that you do have a test coming up on that, so study for it, but I do have to get these videos out for the rest of this book. So, Chapter 7, Section 1 is the cell discovering theory. This is where we invented microscopes to help us discover cells, and after that we developed the theory that to be alive you have to be considered to have cells. So, let's get into this. If you look at these again, those are your teaks. We're getting more into the matter, the big stuff. So, central questions: how are the advances in microscope technology related to discoveries about cells? What are the similarities and differences between compound light microscopes and electron microscopes? And then, what are the principles of cell theory? What are the differences between prokaryotic cells and eukaryotic cells? So we're going to try to answer these questions as we go on. These are our vocab words. Make sure you're getting those done. I am going to check those. But as we go in, hopefully my music's not too overpowering. History of cell theory. See, once upon a time, we had no idea what cells were. And then a really cool guy came by and he discovered what cells are. We're going to talk about him a little bit in class. It's Robert Hooke. He looked at a cork and saw it. Hey, this cork's got cells. And he defined cells like he actually thought like a cell in a prison, like a cell where one thing is alive in. So he said this is cells as well. So a cell is the basic structural and functional unit of all living organisms. Cells are so small that their existence was unknown before the invention of the microscope. Cell theory is one of the fundamental ideas in modern biology. It includes three principles. First, all living things are composed of one or more cells. One especially, starting there. And then cells are the basic unit of structure and organization in all living things. And then cells arise only from previously existing cells, which cells passing copies of their genetic material onto their daughter cells. We've discovered them by looking at our different microscopes. Now we're going to try to have a lab and microscopes just to practice, to show you guys how to focus. And then once we get to where we can use that, we'll start looking at our own cells, look at some cheek cells. We might even try to look at some hair, look at some different stuff to really discover what a cell is. So we have a compound light microscope. This is what we'll use in class. It uses a series of glass lenses and visible light to magnify images. Now, it will magnify up to around 1,000 times actual size. Light microscopes get up to, I think, about a max of 1,500 times size. At that point, you have to use an oil immersion lens. We won't be doing that. But after that, light is refracted so bad that it really, you can't look at anything really clear anymore. So we moved from a compound microscope to an electron microscope, where we can go, instead of 1,000 to 1,500, we can go all the way up to 500,000 times. That's huge. We're going to look at some really cool images of cells that really help us understand what a cell is. Now realize, cells, when we talk about cells, when I ask, what runs a cell? What's the holder of all information? Don't say brain. That's not a cell. That's a conglomerate of cells that made of tissue that made an organ. Do not say cells on, do not say brain as that, all right? We need to make sure that's understood now before we get anywhere else. How does a microscope, electron microscope work? Sorry, tangent. We create an image by illuminating the sample with a beam of electrons. We actually shoot the electrons at it, and electrons hit it, and then they reflect back from the sample, and that's how we image it. Notice if we're shooting electrons, that's pure radiation. Since it's pure radiation and that's electrons, whatever we're looking at, it is in a vacuum, which means there's nothing in the way. It's absolute empty. And then it's like space. It's absolutely empty. And then it will be actually bombarded with that material. So nothing can live in that. It has to be dead to observe it there. Versus us, when we look in the microscope compound, we can see actual living things in that microscope. So we're not going to watch this video. We're going to play with it and observe these different cell types. Basic cells, though, all cells have a basic setup, such as one, they have this plasma membrane. Every single cell in existence has a plasma membrane. That is the bubble, like a balloon. When you blow up a balloon, it has that coating. We could call that plasma membrane. It locks it in, okay? You have a plasma membrane, then you have your genetic material of some form, And then you have something that will break down other molecules to generate energy. Typically, all of that would be ribosomes, which are proteins, and that's the basic setup: plasma membrane, genetic material, and ribosomes, which help generate energy, break down things, and store energy and things of that nature. Now, we have two types of cells. We have eukaryotic and prokaryotic. Basic cell types for eukaryotes, we have plasma membrane, genetic material, ribosomes, but also, see, they're more complicated. We have a nucleus, which is going to take care of, it's a second layer inside the cell. It's going to protect that DNA, protect that genetic material that we don't want to damage. And then on top of that, we have specialized organelles, like your Golgi bodies, your endoplasmic reticulums. We're going to walk through these. You're going to know what they do. Mitochondria, lysosomes, your vacuoles, bunches of different things. You've got to study those organelles, and you've got to memorize them for your tests that you will have later, okay? But that nucleus is the distinctive central organelle that contains the cell's genetic material. And on most of the time, a eukaryotic cell is much, much larger than that prokaryotic cell. Prokaryotic cells don't have any of that stuff. They only have the basics. They have no nucleus. They have no other membrane-bound organelles. All they got, genetic material, ribosomes for energy, plasma membrane. That's all a prokaryotic cell has. When you look at an image, you should be able to see a distinctive difference between the two. It's not something that should confuse you. If you see organelles inside or a nucleus, bam, automatically eukaryotic. If you see none of that, it's just DNA, cytoplasm, which we'll learn about. It's just the goo inside. Ribosomes and a plasma membrane, then that's a prokaryotic cell. They're probably similar to the first organisms that lived on Earth. We'll get into that. Listening to my music. Waiting for 8th period, by the way. Not 8th, 9th period, by the way. It is 8th period. It's my conference period. So it's kind of interesting. Just chilling here. Just waiting for the PowerPoint thingy to load. Sometimes it takes a little time. Oh, there it is. All right. So cell types, origin of cell diversity, eukaryotic cells, they may have evolved from prokaryotic cells. This is where we're bringing a theory. You need to study this theory. You need to memorize this theory because I'm going to ask a question on it. Period. It's called the endosymbiont theory or endosymbiotic. So if you want to spell endosymbiotic, it'd be B at the end, that B-I-O. It'd be biotic, T-I-C theory. Same thing. Proposes that there was a symbiotic relationship that formed between two prokaryotic cells, one of which lives inside the other. Eventually, a symbiotic relationship led to two cells becoming one. Now I'm going to tell you about endosymbiotic theory. So it's not here. You need to write it down. I don't know if it's on the next slide. I can't remember, but I'm going to tell you about it now. The reason why we believe that endosymbiotic theory occurred is because we have an organelle inside of a cell called a mitochondria. That mitochondria, when we look at it, it actually has DNA. Now, every one of our cells is eukaryotic. So we have the cell, and then inside of our cell we have a nucleus, and then inside the rest of the other organelles within our cell is one of them is a mitochondria. It's the powerhouse. Now, if you look at that mitochondria, it has DNA in it. Wait a minute. Wait. DNA is supposed to be where? In the nucleus. Why does the mitochondria have DNA? The reason why it has DNA is because it's not the same as our DNA in our nucleus. It's different. And in fact, it shows the characteristics of a prokaryotic cell's DNA. It's circular. It has no other organelles in it. It has ribosomes packed inside that help make energy. It has circular DNA inside of it, and it has a plasma membrane. It has all the characteristics of a prokaryotic cell. So that's where the theory of one of the parts of theory of endosymbiotic theory came from, is the fact that mitochondria have that. On the other side, we have plants. Plants have chloroplasts, and those chloroplasts are the same concepts. Chloroplasts have actual DNA within them. So we believe that's endosymbiotic theory. Because of those cells have all these other organelles, and think about it, if a prokaryotic cell took in a bunch of other prokaryotic cells and they all started working together, wouldn't that cell get bigger, the one that took everything else in? And it did. So typically they're bigger and they have more specialized functions. And this leads to cell diversity, which also leads to organismal diversity. That's the end of section 1, cell theory. Now, if you look in the book, there's so much more. Make sure you read your book and you do your science notebook. Oh, crap. I lost my all right, I'll get them on in just a second for the next video. But make sure you read that book. Make sure you get that information because you need to know it. Okay, we're not going to cover every little bit. You need to get every bit you can. All right, thank you for watching. Peace! Peace!